Okay, so this is me taking a stab at meiosis. And basically what you're looking at is a cell. All the white is considered to be a cell, which is enclosed in a cell membrane. It has organelles and cytoplasm and all the other stuff that you would find other than the nucleus, which the nucleus is what encompasses all of these chromosomes, which I have 6 chromosomes there. An adult cell has 46 chromosomes, but I can't fit 46, so we're going to work with 6. So in meiosis, there's 2 different phases. There's meiosis 1 and meiosis 2. So I'm just going to work through both of them. In meiosis 1, we have broken down into other stages. The first one is called interphase 1, and that is when the chromosomes start to gather together. They become less spaghetti noodle-like. They get more wrapped up, more formatted, and they duplicate at the end of interphase 1. So each of these will get a buddy and will become a homologous pair, which is this set of 2 chromosomes that we have going on. So basically in the adult, it goes from 46 chromosomes to 96, or I'm sorry, to 92. Ours went from 6 to 12. Yeah, 12. So it's just a lower number for simplifying. So then in prophase 1, which is still part of meiosis 1, the chromosomes have doubled and they are possibly going to cross over, which crossing over is when the sister chromatids, which is each half of these homologous pairs, which are are connected at this centromere. Crossing over is when they can switch feet, if you will, and basically get a combo of the 2. So mine aren't going to show that, but basically what could happen is one of these greens could send some of his stuff from this little foot over to that foot, and this foot would switch. So they would end up switching, and this one would have some of the information originally on this, and this one would have original information from this one. So then what they're going to do is we're going to move into prometaphase 1, which is when the spindle fibers that are going to come from these sides are going to grab onto these at the centromere and align them along the center. So they're going to get aligned to where each of the homologous pairs are on one side. And so what they're going to do is they're going to separate the pairs as a whole. So spindle fiber is going to come on, grab the centromere, and in anaphase 1, the spindle fibers are going to shorten and they're going to grab these. And so each side is going to get one of each of these pairs. So then that moves us into telophase 1, which is when, or through cytokinesis, these are going to move apart and they're going to create 2 daughter cells. So we've got 2 daughter cells with identical chromosomes, homologous chromosome pairs, and cytokinesis is going to pinch them apart, and they're going to be 2 completely separate cells. And these are called daughter cells. So the microtubules have disappeared, and we have our 2 daughter cells. So now we're going to be moving into meiosis 2, which is basically the same thing is going to happen with one difference. So I'm going to show this one more up close and just know that the same thing is happening with our other cell up there. So just like in the first one, this is going to happen. So we've got the 2 cells that are formed in an interphase 2. They're already hooked, like they're already together. And so what's going to happen is now that there's, okay, so we have 6, but again, in an adult cell, it's 46 chromosomes. So what's going to happen is the microtubules are going to come in and they're going to grab in metaphase 2, they're going to grab at these centromeres and separate the sister chromatids. Now, I explained it in the mitosis video, but I want to explain it again in this video. These don't separate in real life like this. They separate down the midline. So that is one sister chromatid, that is one sister chromatid. So in real life, they would be cut from top to bottom. But for art's sake, I have to separate them like this. But anyway, so in anaphase 2, they're gonna get separated by the spindle fibers grabbing at the centromere and shortening, and they're gonna pull them like that. So then we've got what might not be identical pairs because of the crossing over. Some of these could have, like we said earlier, in the crossing over, this one might have information from this one, and this one might have information from this one. So if they crossed over, then they're not exactly the same, but generally they have the same kind of stuff. Like these are still probably for eyes, and these are for hair, and these are for height, or whatever you might want to call it. But they're not going to be identical, which is how kids end up each looking different, because the cells that are created in meiosis are the traits and the genes that are going to be transferred into kids from their parents, and so they might end up different, which is evident in our differences through crossing over, creating these different forms. So what's going to happen next is, now that these have pulled apart in anaphase 2, telophase 2 is going to set in. And so just like in the original one, they're going to pull apart, and cytokinesis is going to split them, and they're going to pinch, and as they pinch, they're going to form 2 new cells. So the same thing, which we didn't show, but the same thing happened up here. So we've got one of the orange ones got pulled over, one of the yellow ones got pulled over, and one of the green ones got pulled over. So we have 4 cells with different but similar chromosomes in them. So these are what will become egg cells or sperm cells. This is how it's created. And they each have only 23 chromosomes, because together, whenever the daughter cells were created, and we only had 2, those each had 46. So when these split, they each get 23. And each of these, as they crossed over, as we said before, are going to be a little bit different. All these little tetrads, they all have different possibilities of getting different genes, which creates different combinations for each child, which is why I have red hair, my sister has brown hair, and my mom has blonde hair. It's all based on dominant and recessive genes and all of that stuff, which I'm not going to get into because I'm just focusing on meiosis and cell division, but these are my 4 egg cells or sperm cells. So I hope that wasn't too confusing.